Click for Biology, Syllabus Statements 5.3.2 and 5.3.3. Looking at the sigmoidal population growth curve. Let's begin with the idea of having a bacterial cell, and the bacterial cell is going to divide by binary fission to produce two cells, and in turn these will divide to produce a population of 4 cells, and this process will carry on over time. On the graph, on the horizontal x-axis, we have time or generations. On the y-axis, we have the population, the number of individuals that we're counting. We also have to imagine that these bacteria are in a restricted environment, perhaps thinking of this in terms of being present in a glucose solution to provide nutrient. Characteristically, the population, if measured over time, would produce data points with this pattern. Steep increasing population, making a gradual transition to a horizontal plateau. This gives us a curve like so, where this would be known as the exponential phase, this would be known as the transition stage, and this would be our plateau population. The exponential growth phase is because at this phase there are no restrictions on the population growth. There is adequate nutrient and space for the population to increase. As the nutrient begins to decline, the glucose solution in this case runs out, as space becomes a limiting factor, and perhaps as the population may produce metabolic waste that poisons the population, the rate of population growth decreases. So the transition stage, the transitional phase, is a slowing in the rate of population increase. Finally, the population will reach a horizontal plateau. This is equal to the maximum population that the environment can support, and is sometimes called the carrying capacity. So the environment would carry a population of this size, which we could see on the y-axis, and this would be the carrying capacity of the environment that we're studying. Notice that in the model we begin with a population of a very small size which expands into the particular environment that we're studying.